Okay, once again, hello everyone. We are live. Let's get started. Thank you guys for being here. Really appreciate everyone. We have a huge, very, very full room. Kind of intimidating, can't lie. Hope you guys are settled in. We have a pretty difficult lecture to give today. This is notoriously one of my least favorite chapters to teach. Number one, because there's a lot of content. Number two, I struggled with it when I was a student. And number three, it just kind of feels like me lecturing about something I don't truly know about. And that'll become evident as we go on. This is chapter 2, general biology, reproduction. Every large thing is made up out of what? Small parts. Every large thing. Rocks, carpets, people, anything. Every large thing is made up out of small parts. And if you look back in time, there was a point in time where religion, philosophy, science sort of all came together, and you ask the question, like, what are we? And from a reductive perspective, it seems easy to say that we're just human beings. But then you look inside human beings when they died, right, or when they're alive and they got hurt, and you see there's things inside of us. So we're not just like these empty cases of flesh that move around, right? So there are organs and tissue beds and muscles and this and that and blah, blah, whatever it is. And when we eat, the things just disappear, right? Where does it all go? What happens? So the question becomes like, you are a human, but you are also a brain and a heart and skin and eyes. And in the eyes are proteins, and in the proteins are atoms. And it's like, how far does this go, really? And in order for reproduction to occur, you can't just reproduce one thing and nothing else, right? Some things get produced, some things get reproduced, and some things get constantly destroyed and constantly recycled. And this happens throughout the body, right? Let me clue you into something. Your heart, the cells in your heart, the cardiac myocytes, right? Be okay? Am I like? Okay. Your cardiac myocytes, right? If they die, let's say you had a heart attack. By the way, what's a heart attack? Does anyone know? Anyone know how a heart attack works?
When blood stops flowing.
Yeah, but why? Your heart, let's take the box model of the heart. Your heart is split up into 4 chambers, and there are arteries that run along here in order to give your heart the circulation that one wraps around behind, right? So they're sort of these arteries that come like this, and over time, because of diet and stress and smoking and this and that and blah, 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 you get a little bit of a blockage, and that blockage can turn into complete blockage, and then that will kill the oxygen supply downstream from that artery, which will choke out those cells, lead to inflammation. Those cells will die, right? And eventually they will scar over. When you get cut, you scar over pretty well on your skin, because your skin is capable of reproducing skin cells. I have a very large cut on my leg right here from when I was running track and field. A track spike went right through my leg during a race. But it scarred over pretty well. I have a keloid scar there, right? It's like a hypertrophic scar, right? But it did scar over, and I still have mobility, and I can still like sort of feel the part around my leg and things like that. But when your heart scars over, it doesn't really regain function at any point. Why is that? It's because heart cells, and this is something that we didn't speak about before when we talked about cell types, these are permanent. They're permanent cells. They don't actually replicate, divide, grow in the classic way. The heart cells you have are the heart cells you get. That's it, right? Same thing with what, to a point? 25 years old, right? What do I have that I have for the rest of my life? My brain. Neuronal plasticity is the ability for neurons to actually generate connections between one another. But do neurons duplicate and grow and regenerate? No. Well, I hope not, because there's such a limited, defined region of space inside the skull that if the neurons started dividing, you would actually start to outgrow the space that you have inside your skull. That would lead to an increase in the intracranial pressure, and that comes with a bunch of different problems, right? But there are cells in the body, and the majority of cells in the body, I would be willing to say, that are constantly dividing. And this division that makes up the different tissue beds and organs and systems and blah, 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 whatever it is, this is what we know as cellular reproduction. And it is the cellular reproduction that we must talk about in order to look at the big picture, right? Because almost everything inside of anatomy, physiology, this, that, the other thing, is taught from a perspective where it's like, well, here's the big picture and let's look small. The reason for that is because they assume that you come in with the knowledge of what does the small look like, right? And that's why we started with the cell in the first place, and that's why the chapter before this was the cell, right? By the way, for the people watching online, this chapter gets very nitty-gritty into endocrinology at the end, when we talk about reproduction from the perspective of the menstrual cycle, which is going to be a completely different part of the video because I like to post that by itself because it's so heavy in content. I would heavily suggest that you watch my Fundamentals of Endocrinology lecture, which I will link in the description. And if you can't find it in the description, please just go looking for it. It should be right there. It is imperative that you watch that in order to fully understand everything that's going on in this lecture, as well as chapter 5 of biology, which is the endocrine system. Okay. In order to talk about cellular reproduction, we need to talk about the cell cycle. Cell cycle and mitosis. Correct? This is a cell from my cheek, a cheek cell. Right? Scrape the inside of my cheek with a wand, and then I put it under a microscope, right? If I were to get a strong enough microscope to sort of look at the inside of this cell, and let's just say that it was in the process of replicating, right? Or at any point. It's a cheek cell, right? Does anyone know what sort of cell we call this? What subtype? This is a kind of weird question. But what type of cell do we call this? We have two different types of cells in the body, which have two different numbers of chromosomes within them. The somatic cell, right? A somatic autosomal cell, correct? You'll find that there's a lot of like sort of double names inside of medicine and biology and things like that for whatever reason, bless you. And these cells are diploid. Meaning, diploid, listen very closely, of the necessary genetic material to code for the genome, it has two. Of the necessary genetic material to code for the genome, it has two. We are not talking about the stranding. We are not talking about the way that it does that. We're not talking about what genes are turned on and turned off, just saying that for the necessary completion of the genome, it has two of those copies. Correct. So, in humans, how many chromosomes are needed to code for the genome? 23. 23 chromosomes are needed to code the entire human genome. Am I talking about single double-stranded? No, I am not. 23 chromosomes. And if you get one copy of 23 from your mom and one copy of 23 from your dad, that would make 46. This is what we call the 2n number. The 2n number is 46. Correct? Yes? So when cells exist within the body, and they're not dividing or they're not turning into germ cells, which are sexual cells, which pass on and recombine and create offspring, right? They have how many chromosomes in them? Louder. 23. No. How many? 46. They have two copies of each of the 23. I just told you, somatic cells are diploid. And if we need 23 in order to code for the genome, and diploid means you have double the amount needed to code for the genome, that means that you have 46. Take a fly, for example. Let's say that there's a fly out there who has 2 chromosomes in its entire genome. That fly floats around with 4 chromosomes inside of it. One of pair 1, one of pair 2. I like to do this sort of demonstration. It takes a while, but let's say that you have chromosome 1 and chromosome 2. That's not a good way to show it. Chromosome 1, chromosome 2, and chromosome 3, and chromosome 4, and chromosome 5, blah, 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 all the way down to 23, correct? You have a pair of each one. I have a pair of chromosome 1, and a pair of chromosome 2, and a pair of chromosome 3. One came from my mom, one came from my dad. Does that make sense? We need to lock this in order to understand what's going to happen later, correct? I'm going to leave that there. Eukaryotic cells replicate through the cell cycle, correct? And this is basically a series of phases that the cell goes through where DNA is replicated, split, and the cell divides. You hear me? Yes? Yes? I need validation, guys. I didn't get enough as a child. Come on. Am I in frame? There are much better diagrams of this in your textbook, in the Kaplan textbook. What smart person can tell me what M stands for here? Exactly. There are 2 things you need to remember. The cell is either doing one of 2 things. It's either dividing or it's not dividing. Mitosis is the act of cell division. So everything that is not mitosis is not mitosis. Very simple. We have a name for not mitosis. What is it? Interphase. Very good. Interphase. So we have interphase and we have mitosis. And judging by the diagram, that tells us that G1, S, and G2 are collectively known as? Loud. Come on, guys. There's a lot of you in here. Interphase. Good. Very good. Fantastic. Interphase is the longest part of the cell cycle. It goes through these motions, and interphase, when a cell is not preparing for, when a cell is preparing for division, aside from when it's not doing that, which we'll talk about later, is the longest part of the cell cycle. It prepares for mitosis, and basically that's all it does. By the way, for anyone who's new here, you guys can get up, leave, eat, sleep. This is a very, very free environment. I don't care if you're eating in front of me. Just enjoy the lecture. That's it. That's all I'm here for. I'm here for your comfort and accessibility. All right. Cool. Interphase, like I said, is the longest part of the cell cycle, correct? Cells that are not dividing will exit this cycle and go into, what is G0? It is basically just where the cell hangs out, right? People sometimes, when they're older, they're like, especially doctors do this a lot because they have the liberty to do so. They sort of just like pick up and pack up whenever they want, hopefully, if you have that sort of schedule. They're like, yeah, well, I just quit my last job. They're like, oh, what are you doing now? I was like, nothing. Just chilling, living my life, right? G0 is the cell chilling and living its life, right? Does all the cellular processes, does everything it has to do, does its job to maintain itself, right? But it's just not working towards dividing right now. When you're in G1, S, and G2, you're kind of working towards dividing, correct? And the reason for that distinction is not random. It's because of how important mitosis is, right? Because of how important mitosis is. The entire time leading up to my educational journey to medical school is leading up to what's happening this upcoming fall, applying to residency. Every single one of the past 21 years, whether I knew it or not, was leading up to this fall. That's what it was all leading up to, right? Okay, so it's almost like that. So we can say that the combination of G1, S, and G2 is interphase. And within interphase, individual chromosomes, for example, let's look at the fly again, with 2 chromosomes in its entire genome, correct? Which means that it has a pair of 2 chromosomes each. Sorry, it has 2 pairs of chromosomes, which makes it 4, right? These individual chromosomes are what? Not visible. Because they exist in a less condensed state. They're not actually going to look like that. They're going to be this weird tangled mess of DNA that you're not really going to see inside the cell, correct? And the reason for that, who can sort of like deduce the reason that happens? Why isn't the DNA as tightly wound as it usually is? What part of the cell cycle are we in right now? Interphase. And during interphase, we're preparing for mitosis. And in order to prepare for mitosis, what do we have to do to the DNA? We have to replicate it. You have to make it over again. Imagine you're trying to transcribe a book, right? I made this distinction last time, and I think it's a distinction that doesn't go recognized by a lot of students. The difference between transcription and translation. Anyone here ever had like a teacher for another language, like at home, where they would make you like copy lines of like different, like stories and poems and things like that, and you just copy it. You look at it, you copy it, you look at it, you copy it, right? You've done that before, to train your hand in writing that, right? Transcription is when you look at a language and you copy that same language. But translation is when you go from this language to one that other people understand. That's the difference between transcription and translation. So in DNA synthesis, transcription is actually done in order to transcribe the DNA and replicate it to get ready to divide later. Because the thing is, we want everyone to have the same amount of DNA, right? Let's say that I had a kid, and I had $100,000 saved up for this kid. And I was like, you know what? I'm going to make sure that each of my kids has $100,000. I have another kid. What do I have to do to my money? I got to double it, right? I got to double my money. So if you want every cell to have that same amount of DNA, you got to double your DNA, right? Okay, now, here's the question. If I said 2n equals 46, meaning that the diploid number is 46, we have 46 chromosomes in the body, when that DNA replicates, how many chromosomes are we going to have? Huh? 92 sounds like the right answer. Not really. It's still going to be 46. And we're going to talk about why, right? Remember in the beginning I said I'm not talking about single or double-stranded. I'm talking about the number of chromosomes. Later on when we talk about mitosis, we are going to talk about single and double-stranded. So if this trips you up, we will clear the air. Okay? All right. Cool. Everyone good on this? Any questions? Don't be shy. Promise.
It says not visible, less condensed state.
Yeah, less condensed. So let's say that I held up 100 strands of human hair like this from across a football field. Would you be able to see them? Probably not. But if I took them and I crumpled them up into a ball and I held them on my hand, you might be able to. Maybe. Because they're going to take up a lot of dense space. Correct? Something like that. So the more condensed the DNA is, the better you're actually going to see it through the microscope, correct? Okay, cool. By the way, brothers, it's Thursday night. We're going to dinner. Any questions about the chapter, about life, about your deepest, darkest fears and secrets? Anything? G1. Refer back to that diagram that I hope you drew or copied from the book, right? And if you guys need a professional diagram to mark up, like to put on your iPads, I want you to look up cell cycle Kaplan MCAT. That image will pop up. Cell cycle Kaplan MCAT, right? Is that for me? Yeah. I'll take it in a second, yeah. I would really, really love to, but we're in the middle. So I will forego the pleasure of eating for all of you because you guys mean that much to me. Listen, if someone means more than food to me in my life, y'all are tripping. You don't understand. I'm a big back. Listen, when I say we're going out to dinner every Thursday, y'all better not flake on me, man. And if you do, I'm eating by myself. All right. This beginning of the chapter is a little bit boring because I know that we're kind of split. Like half the people know this really, really well because there's like cellular biology majors and you have done this like 100 times. But promise, stick through, it gets better. And the fact that we're doing this now is actually going to make the end of the chapter a lot easier, right? The MCAT is a lot of patience and waiting. It's a lot of watchful waiting. You just look at yourself. You say, do I know it? Do I know it? Do I know it? And when the hammer comes down, you do, right? Okay. G1 is known as the pre-synthetic gap. The fact that it is known as pre-synthetic clues you into something about the diagram. G1 comes before S. So it's literally just the pre-S. And the G actually refers to growth. So this is pre-synthesis growth phase 1. G1, pre-synthetic gap, right? This is where all of the bulking is done. This is where you're putting on the pounds, right? The cell replicates mitochondria, which have what type of replication? What are the genetics of the mitochondria? Has its own DNA. Has its own DNA given to you by the mom. Every single mitochondria in every single cell in this room can be traced back to one female ancestor. A single one. The first female human ever. Correct? Okay. The mitochondria replicate. You also have the ribosomes, the endoplasmic reticula, etc. All these important organs, right? And because of all of this growth, you get an increase in size. This is the bulking, right? We have about an hour till Maghrib, right? Roughly? Okay. That should be good. And the passage from G1 into S is actually gated. You guys might remember my thing about the gated community. It's like, hey, listen, I know where you live in the suburbs, but you're just not on my level, right? That's what the S phase is saying to G1. It's like, yo, I know you put in the work, you got rich, you got your money, you made sure that everything was done right, but this is a different tax bracket right here, right? There is a restriction point. Are any of you guys married? Anyone? You're married? Okay. When your husband does something, do you double check what he does? Sometimes? 100%, right? And it's not that you don't trust them. He's probably done it 100,000 times for her before, just out of his love for her. Inshallah, may Allah bless them. Say Ameen. Right? He's probably done it a million times. He knows how to do it to the T. But it's not that you don't trust them. It's that you know what can happen if you don't check. You know what could go wrong, right? Oh, patient's labs were fine. We just checked 4 times in the past 24 hours. Check again. 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 Because if you get it wrong here, you're done for. Right? I need you guys to really understand this point. It's critical, absolutely critical that you understand the reason that a restriction point is there. A restriction point is there not because the cell is inefficient, but because it has so many things to do, and it executes them all so perfectly, and there's so much ahead that if one thing is out of place, then we're all done for. Right? There's a process in Islamic jurisprudence studies known as ijtihad. It reflects a serious and concentrated study and focus. There is extreme amounts of diligent prep work that goes into formulating a law. For example, a lot of modern Muslim jurists are asked, what is the ruling on investing in Bitcoin? Is it allowed? Is this something that we have to pay charity on? Is this something that we can put into our wills? Is this something people can inherit from us? Is it allowed in the first place, right? There is an immense amount of ijtihad that goes behind this, a serious study. This is the cell's ijtihad, the deep and serious study of what's happened. And you need to see if we can move forward from there, right? And the reason I'm making such a big point about this is because we're going to see what happens when that thing goes wrong. When this fails. Horrible things. Okay. Are you all with me? Good. It's more of an answer that I like. This restriction point is basically to ensure that the cell has the proper amount, type, and accommodations of/for the DNA. You want to have a kid. Do you have food at home? Do you have money? Can you afford diapers? Do you have somewhere to keep the kid? Do you have good family support? Are you ready? That's the question, right? You want to replicate. You want to make a new cell. You have enough mitochondria, enough ribosomes, enough space, enough DNA. You have all the genes properly matched up. Is there anything wrong? Is this cell going to survive? And more importantly, is this cell that you're about to make going to kill us? That's the question. And who's us? Does the cell know that it's working as part of a whole body? No, not always. It doesn't always know that fact, right? Through hormonal regulation and through communication, it knows that there are other cells around and there are other things going on, but it doesn't always know the fact that it's part of something bigger, right? S phase. Synthesis. So, remember how I said that the G phase, the DNA is open for transcription? Did I say replication? No, I did not. There's a difference between why the DNA is open in the G1 phase and why it's open in the S phase. In the G1 phase, it's open to code for the genes and the proteins that are going to make all of those materials that you're prepping. In the S phase, it's now open to be transcribed and replicated. This is the DNA synthesis phase. Correct? This is where the cell replicates genetic material. Make sense? And this is so that, therefore, each daughter cell contains what? Identical DNA. You guys know how whenever there's someone who scores really well in the MCAT, everyone's just like, what was your study schedule? What did you do? How did you do it? Let me do what you do. So when this cell that's lived its whole life and never had anything go wrong is about to divide, it's giving up exactly what it had. Because they said, it worked for me, it's going to work for you. Identical. Same thing. Same circumstance, same DNA, same look, same habits, same environment, same everything. All the same. Correct? Why wouldn't it work? Correct? Right. I'm going to introduce you guys to a new set of abbreviations I use all the time. I use them everywhere. The A with the arrow and the P with the arrow. This means avant, and this means post. Avant means before, and post means after. So the P with the arrow means after. Correct? Okay. So after replication, each chromosome, how many chromosomes? Human? 46. Consists of 2 blank blank bound by a blank. 2 sister chromatids bound by a centromere. Centromere. You have a chromosome. Top and bottom. A chromosome. That in the G1 phase was open and replicating and doing whatever it had to do. When it enters the S phase, let's say the S phase occurs, it happens to it. This G1 chromosome that was single-stranded now looks like this. And it's not that those are crossing over the centromere. They're pinched together, right? So this is one side, and this is the other. Can you guys see that? You're taking this, you're taking another, you're pinching them together. So this is one of them, and this is the other one. They're pinched at the center, correct? And they're held together like glue with the centromere. Does that make sense? Are you guys with me? Are you following? Because it's about to get very complicated.
So the curved arrow in the
S phase happens.
okay, gotcha.
Yeah, S phase happens. You guys all right? Good. So these are your sister chromatids. And I want to include these right here because I don't want you to get confused. Sister chromatids. Correct? Yes? Good. Copy that down. It will take a second, we'll move on. This is very slow, very boring stuff. I know you guys probably aren't having fun right now. You will. In just a few short minutes, you'll be having a lot of fun, too much fun even. Yes? Sam, how are you doing? Good. You good? Okay, perfect. Can I erase this? Yes? Yes. So G1, let's just say that G1 happened, the S phase happened, and then that got spat out. G1 happened, S phase happened, now that got spat out. And not even that you would actually see it in G1. It's not going to look like that in G1. I'm just showing you that it's single-stranded. Does anyone remember the name of the proteins that DNA wraps around in order to tighten up? Histones, which I previously mistakenly said contain histidine, but they actually contain the other two basic amino acids, lysine and arginine. Very good. You guys are smart. Smarter than I was. Can I erase this? So now we have sister chromatids. Now what? I'm going to tell you guys something that I'm going to say once, and I will take no questions about it. Because when people started asking questions, then I started to mess up. I'm going to say this one time. And for the people it clicks with, and for the people it doesn't, it doesn't. In the G1 phase, we have 46 chromosomes that are single-stranded. After the S phase, we have 46 chromosomes that are double-stranded. That means individual DNA chromatids, we have 92. Individual. And I got into a heated argument with a student because and I stopped doing this, but I once called this 4N. And I stand by that. I stand by myself calling that 4N. Because if N is 23 single-stranded and 2N is 46 single-stranded, then 4N would be 46 double-stranded. You're doubling it. I'm talking about the number of specific DNA chromatids. 92 from 46 double-stranded. Does that make sense? Okay. I'm not going to answer questions about that. If it clicked for you, it clicked for you. If it didn't, it didn't. It's okay. It's not an important point. Welcome. How are you guys? Doing okay. From S to G2, we have another restriction point. Do you think that this restriction point is easier or harder to get through than the last one? Why? Why harder? Much more difficult to get through than the last one. Every single time you take an exam, right? You take the MCAT, you take step 1, you take step 2, you take all the shelf exams, you do this, you do that. As you get closer and closer and closer, the exams become more critical. They're twisting the knife deeper into your head, being, no, answer, answer, answer. This is the question, answer. Now that you've replicated the DNA, anything could have happened there. Anything could have happened there. When you replicated the DNA, you could have swapped a C for an A, and now we're all screwed. Everyone's screwed. There is a scrutinizing, incredibly difficult position to get through right there that is much more difficult than the last. As difficult as the last one was, this one's harder. Correct? So the G2 stage occurs after and we call it SG2 checkpoint. Correct? Yes? How am I doing? Good. I mean that seriously, by the way. Occurs after the SG2 checkpoint. So the DNA has been duplicated. Have you guys ever noticed how I write the word the? It's like one big stroke. That, to me, that says the. My mom saw me do that one time. She's like reading something I wrote in philosophy, and I wrote the word the, and I told her, yeah, the. You know, there's your T, your H, and your E. The. My friends describe my handwriting as a mix of script and garbage. And I say it's almost as though you took a spider dipped its legs in ink, and let it run over the paper. I hope you guys tell me when I should rewrite or re-explain something. Okay? I am not a perfect individual. I might be very close, but I'm not perfect. Okay. And the cell here, the cell has a job. It's going to check the house. Like I said, when you're about to have a child, right, you're going to check the house. You're going to check the heating, check the walls, check to see if there's lead in the paint, check the room, check the money, check the bank account, check the food, check the this, check the that, make sure that people are on standby in case you need them, right? Checking everything. Organelles, DNA, space, this, that, membranes, vesicles, everything. You're checking everything. You're doing your whole mental checklist. Anyone here ever moved houses before? Not like for college, but like actually moved homes. It's hard. It's really difficult. It's really difficult. And as a human being, you're always going to forget something. Always, every single time. What did I forget back in New York when I came to Dallas? I forgot my running shoes. I had to buy a new $200 pair of running shoes. Annoying, right? But the cell doesn't forget. Definitely not purposeless. Not without purpose.
So when it goes to the first checkpoint,
Mhm.
can it forget things, or is it just this one?
I want you to think that the cell is always capable of making mistakes. It just doesn't really happen all too frequently. And what I mean by all too frequently, I mean 1 in a billion trillion times. It'll happen. And then when it does make a mistake, it's caught by something else. Finally, after the G2 phase, we've checked the house, everything's ready, all the synthesis is done, restriction points have been passed, this, that. What do you think it's time for? It's time to party. I have a habit, I don't know why, it's just like a sort of little ritual of mine. I never erase the title of the chapter, unless I need the whole board, right? In which case you will see me dramatically wipe off the title of the chapter. Very dramatic. I love the drama. I'm with all the drama. If you guys like being dramatic, do it. Mitosis. This is the M phase. You guys are always laughing about something.
So if the DNA is replicating in S phase, but it's also connecting by the centromere, does that mean the cell's also actively working to continuously make the proteins needed to attach the chromosomes?
It's a very good question. The question was, if the cell is constantly replicating the DNA in the S phase and attaches them by the centromere, which is a protein or something that the cell needs to make, does that also mean that while replicating, it is creating proteins and doing this and doing that? If the cell, let's take this for a ride. If the cell wasn't producing proteins at the time, what would happen? All of the cell functioning would actually halt. You're still having transcription factors come in, hormones, signaling, this, that, the other thing. The cell can't stop for anything. So even though there is DNA synthesis going on, there is still DNA activity. And that activity is producing a lot of changes at the cellular level that are helping to maintain the cellular and physiologic homeostasis. So yes, both things are happening, to the best of my knowledge. Mitosis is split up into 4 phases. We have prophase, metaphase, anaphase, and telophase, or telophase. Would you guys like me to speed up? Yes? A general consensus of yes? If one person says no, I'm not speeding up. Say no, or just shake your head no if you don't want me to. I'm not going to out you. Okay. I got the message I needed. Prophase is the condensation of genetic material. And this is where you have the visibility of the chromosomes. So you actually won't be able to see them until you get to the point where they're just about to divide. You won't even be able to see them until you get right here. And this is also where you have centrioles. Anyone remember what centrioles are? They're those little protein things that move to the opposite poles of the cell while things are going on, and they're going to sort of throw those fibers to pull the chromosomes apart later, right? Okay, so the centrioles form and they start to move away, right? In metaphase, chromosomes, and sometimes I just like to remind people that chromosomes look like this right now, right? Chromosomes line up at the center of the cell. Does anyone know what that's called? I know this is called an ampersand. But what's that called? Is it just an @ sign? I don't know. If there's a name for that, someone find out and tell me. Not right now. And the center of the cell actually has a name. It's called the what? It's named after the part of the cell cycle that it's in. Part of mitosis. It's called the metaphase plate. Metaphase plate. The metaphase plate, also known as the equatorial plate, because it's going to split the cell in half. Audio, video, we're okay? Yes? Perfect. And the centrioles that we formed before now are at opposite poles of the cell. Can I draw over here?
Yeah.
Okay.
Where your head is right now is probably where you should stop drawing.
Right here?
Yeah.
Okay. Prophase, we can physically see the chromosomes. And we have these little things called centrioles, which are later going to play a very big role, that are starting to move to the opposite sides of the cell, while the chromosomes move to the middle. This is prophase. Metaphase, right? The chromosomes now line up in the middle of the cell at the equator, the equatorial or metaphase plate. And these centrioles are at the edge of the cell. You guys all know how this story ends, right? We're going over it because we're actually going to talk about this next time as well. Yes?
Could you re-explain the first picture you made?
A little louder.
Could you re-explain the first picture?
Yes. Do you remember how in the cell there was a nucleus? We said goodbye to the nucleus, and the chromosomes are exposed. That's one thing I forgot to mention. Before prophase, in preparation for it, the nucleus has to dissolve. That whole nuclear membrane, that double envelope, rips apart. And I can't believe I forgot to mention that, because that is something the MCAT loves to harp on, that the nuclear envelope has to disappear in order for mitosis to take place. Please write that down. That the nuclear envelope must disappear before mitosis takes place. I saw your brother yesterday. I was looking for him after the talk, but I couldn't find him. I was gonna tell him that we met. In anaphase, these sister chromatids separate. How does that happen? These centrioles throw things called, you guys know it. You guys heard 100 times, loud. Spindle fibers. I like to draw it the proper way, because it actually shows the animation of it going away. You pulled that way. Pulled this way. Boom and boom. To opposite ends of the cell. Correct? And just for completion's sake, anyone here from Boston? Or ever lived in Boston? Ever? Yeah? My sister left to Boston at the time that I was studying like 9th grade biology. So I used to send her this picture. It was like, oh my God, it's you and me. We were like going in opposite ways. My sister lived in Boston for 11 years. She loves that place. I love that city too. Boston's a great city. Boston's a really nice city. Chicago, Boston, New York City, Dallas, San Diego, great cities. Anyone here from like a weird city? Like nobody's ever heard of this place before? Huh? Where are you from? Never heard of it. I did one time pray on a local, like sort of highway road-ish area in Arkansas in the middle of the night because it was like Fajr time was running out and it was super cloudy. And we were driving down this highway back from Dallas to New York. Me and my friends were driving back to New York. The first time I visited Dallas. And there's a photo of me with my hands wrapped in prayer, and you can just see like the hill underneath me with like all the fog while I'm praying, because we took turns praying because we were just like surrounded by Arkansonians, and we had no idea whether we were going to get shot or not for being in the middle of the road praying. And the thing is, like, we were just coming back from Dallas, so we were like thobed up. I've got like my Dean over Dunya hoodie on. Like, I was a prime suspect at the moment. All right. And that's basically the only thing that's important about anaphase. Those spindle fibers get thrown. Like I said, these are called spindle fibers that come from the centrioles. Right? You have these spindle fibers that come from the centrioles, correct? Anyone know what spindle fibers are made of? Microtubules. Yes, that's also an MCAT fact. That these spindle fibers are actually made of microtubules. And another MCAT fact, what are microtubules made of? Tubulin protein. Very good. So the spindle fibers in the centrioles are made of microtubules, which are made of tubulin. Right? And this is why I introduced the idea of what did we come from in the beginning. You break it down and break it down, break it down, and break it down. And after tubulin, you have to get into the amino acid chain, then you have to get into the DNA and blah, 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 and that goes in a cycle, right? Okay. And finally, for telophase, the spindle apparatus disappears. The nuclear membrane reforms. The chromosomes uncoil. Yes? I like the nods of approval. these guys, I'm just going to put a little bit of haze on them. As chromosomes start to sort of disappear, right? The membrane starts to involute inwards, correct? You have a sort of pattern, huh?
What's the word next to chromosome?
Uncoil. Script plus garbage, right? You have a little sort of thing going down the middle that I'm going to ask you to name in a second. And the nuclear envelope is beginning to return. And you actually no longer see that spindle apparatus that we spoke about before. Take a picture if you need this for future reference. I'm sure you don't, but. Nah, shut up. Does anyone need to take a picture of that? I feel like we know this pretty well. Okay, cool. What is that called in the middle? It's called a cleavage furrow. That's where the cell is going to split from, right? The cleavage of the cell happens from there. This process of the cleavage of the cell and into two different cells is known as cytokinesis. Cytokinesis, correct? And this makes two cells which are both 2N single-stranded, which is exactly what we started with. We have successfully replicated, moved, split, and divided the genetic material inside the cell. Correct? And this is all within what phase of the cell cycle? The M phase. The M phase. So G1, G2S was just to get, G1S, G2 was just to get ready for this. That was it. Understood? Good. If you guys understand this, then knowing meiosis actually won't be that difficult. Because now, I feel like in previous years, I spent a long time going over meiosis when I could have just said one or two things, and it would have been clear. There are rules to meiosis. Number one, it starts at the same place. Starts at the same place. Now we are talking about meiosis. What is pause, reel it back. What is meiosis? Meiosis is the production of haploid cells. If diploid is 46, haploid is 23. Enough to code the genome. I want you to remember that very clearly in your head. Enough to code the genome. The whole genome is coded inside that cell. Can the cell grow by itself? No. This cell cannot turn into an organism by itself. For reasons you will learn in embryology specifically, you need 2 copies of the chromosomes in order to sustain life. What happens if you have 3 copies of all the chromosomes? That is something known as a molar pregnancy. Very scary stuff. You can look it up. There are partial moles and complete moles and blah, 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 and this and that, and it's creepy. A haploid cell, 23, single-stranded. Right? I'll make the distinction single-stranded. Right? Why would we want to make haploid cells? So that when you combine 2 haploid cells, you get a diploid cell. And that diploid cell can undergo what? Mitosis, not meiosis. That diploid cell can undergo mitosis and divide. and divide and divide and divide and turn into a organism. Turns into an embryo and a fetus and a baby and an organism and an adult. We came from one cell, all of us, everyone in the room, right? Unless one of you is a robot. I would not be surprised. Suspicious. Right? Okay. The production of haploid cells. So let's take a look at this for one moment with me. I know it's so boring, but I need to drill these concepts into your head early on so you don't get lost later. Right? Look at this. Let's go back to our example. The 2 chromosomes, that's the diploid number, right? The haploid number would be 1. Haploid number would be 1. And if you were to combine these two, you would get a cell that is capable of sustaining life. The haploid number is how much you need to code the genome. The diploid number is how much you need to sustain life. That's a very important distinction. You guys feel me? Hopefully not literally. That'd be weird. Unless you're that guy. Or his brother. By the way, I have to have dinner with the 2 of you soon. Meiosis is split into 2 parts. Correct? Because we have the same starting point, right? So let's take a look at our 2 diploid chromosome, correct? Start at the same place. Remember how it looked like this? Yes? How can you get that to look like this in one go? You can't. You can't do one divisive move and make that look like that. It has to be 2. Either you split these apart and then split the sisters apart, or you split the sisters apart and then split those apart. Correct? Which one do we do? Let me define these for you. These 2 chromosomes, I'm colorblind. Okay? My 23rd pair of chromosomes has a defect. I have an X chromosome and a Y chromosome. My Y chromosome has a defect, right? Maybe I'm not the best example of this, but let's assume that these 2 were quote-unquote the same. What would you call these 2 chromosomes with respect to one another? They're part of the same pair, which means they are homologous. Let's take a look at my 13th pair of chromosomes. One of them looks like that, and one of them looks like this. These 2 are considered, I just said it, homologous chromosomes. Now, take my 13th and divide it after S phase in preparation for mitosis. These 2 are considered still homologous chromosomes. The number doesn't matter on either of them. They code for the same things on the same pair with the same genes from different parents. Those are homologous chromosomes. So my question to you is, there's 2 steps of meiosis, like I said. Question, do we separate the homologous chromosomes first, or do we separate the sisters first? Homologous chromosomes. Separating siblings is difficult. Separating siblings is difficult, right? Okay, so we're going to look into that as we continue to do this. And that is a fact. If you didn't know that about me, I am colorblind. I'll explain the genetics of how that works at the end of the lecture because it's important. You have to know it. It has a very specific inheritance pattern that you must know. Does anyone know who had to be colorblind for me to be colorblind? Is my mom colorblind? No, my mom's dad is colorblind. Figure that one out in your head. My mom's dad is colorblind, so I'm colorblind. But neither of my sisters are. Because who is not colorblind? My dad. starts at the same place. Number 2, it's 2 events, back to back. Number 3, guys, creates haploid cells. For humans, that's 23. And number 4, we dance. We do a dance in the middle of meiosis. You gotta go? I need to tell you guys about the dance. I will demonstrate the dance. No, I will not. No way. No shot. All right? The dance is called what? Who knows what I'm referring to? What's the dance? Crossing over. Crossing over is the dance. Can I erase this? Okay. The stage of meiosis that you're in is denoted by a number. And the stages actually have the same steps as mitosis, correct? So there is meiosis 1 and meiosis 2. I always thought meiosis was spelled kind of weird. Would have thought it was meiosis, but that means something else in medicine. Right? What does myo mean? M-Y-O? Muscle. Good. What does myositis mean? Inflammation of the muscle. Right? Good. Infection was a good guess. Who said that? Muscle infections are nasty. Disgusting things. Is anyone not queasy and want to see like something? Does anyone like looking up like really gross medical stuff? I want you to look up Fournier gangrene. Fournier gangrene. Make sure you turn off your safe search. Meiosis 1 starts with prophase 1. Shouldn't have said that right now.
You know, there's an NBA player called Evan Fournier, so whenever somebody searches his name up and clicks on images, that's what
Oh, that's terrible.
that's like a running joke.
That's awful. If you guys need to wash up for prayer, I suggest you do it now. We're going to stop in just a little bit, and also this part of the lecture gets a little bit repetitive. It's just a little bit of a difference from whatever it is. Actually, after I'm done with crossing over, then go. Prophase 1 is the same thing as prophase and mitosis, except mitosis prophase plus crossing over. What was mitosis prophase? The nuclear envelope disappears, the chromosomes become visible, these move to different sides, the centrioles, right? Everything, all of that happens. Crossing over occurs. Guys, this is the part of the lecture that I really need you to dial in. This is when things starts getting interesting. Because this specific phenomenon describes all of genetic variation amongst human beings that isn't described by spontaneous mutation, right? Every single difference that you see between generations, the reason that I don't look like a carbon copy or mix of half my parents and half this, half that, is because of crossing over, right? And it is a point at which problems can happen. And if problems can happen, the USMLE will ask you about it. That's the important part here. I have a cell. Correct? I have a cell. Prophase. This is the prophase of mitosis. Right? as these cells begin to move towards as the chromosomes begin to move towards the center, correct? You guys know how those are homologous? They're the same pair, part of the same thing. One's from mom, one's from dad, right? My mom has deep, dark black eyes. My dad has very light brown eyes, right? There's not going to be any other color. I'm Bengali, right? I know one Bengali guy with green eyes. Handsome man, very handsome man, right? Let's zoom into these for a second, right? I want you to consider them like blocks. I'm going to shade in this domain. And on this one, I'm going to shade in everything except that domain. On that domain, I'm going to put some dots, not ribosomes. Crossing over is the phenomenon by which these homologous chromosomes kiss. Mom, dad. The homologous chromosomes kiss. And what do? They swap a tiny bit of complementary genetic material, meaning that something like that takes place. You guys see what happened? You guys see that, right? What I'm going to do, I think it looks better if I erase this, right? Because now you can see that it's different, right? You guys see how that happened? Why did I say that this impacts all of the genetic variation? Because what's going to happen to each of these? This is 1, 2, 3, 4 different things inside of one cell. All of those are going to go 4 different ways, right? Let's say I happen to pick up number 2. Guys, lock into this. If I pick up number 2 before crossing over, I get straight mom, right? That's it. Everything that's in my mom, I get. But because of crossing over, I get 80% of my mom and just 20% of my dad. And then when I grow up and I have a child, right? Yes? That's one of my chromosomes, but there's another one of those too, right? Inside of me. This is happening inside of one person, one of my parents, from his mom and his dad. Let's say the same thing happens in my mom, right? From her mom and her dad. And those two things mix. Now I have 4 people's DNA inside of me, and this same thing happens again in me. So what was 80% my mom's mom and 20% my mom's dad now becomes 80% my mom and 20% my dad, and it keeps going and going and going and going and going. And that's how genetic variation happens. You guys see this? This happens in my parents. Then it happens in me. Then it happens in my kid. Then it happens in their kids. And you get a mix of thousands of different people of genetic material in one single cell. That's how it happens. Do you guys kind of see it? It's like building blocks, right? You guys get that? If you don't get it, there are beautiful animations online that do a very good job of showing how crossing over works. One thing that I did to sort of show it is homologous chromosomes. Crossing over. Homologous chromosomes. Crossing over. But this one came from there. And this one came from here. Meaning that every single time you swap over, it's coming from a different place. Does that make sense? Because once you swap, it's not like you're the same chromosome anymore. You're not 100% from one person. Does that make sense? It's a little bit difficult to wrap your head around if you don't see it, if you don't physically see it in front of you. I might do a demonstration. I might make something to do a demonstration with. Okay, The crossing over happens in prophase 1. Yes? In prophase 1. After that, it is business as usual. Metaphase 1 is the same. Anaphase 1 is the same, but the homologous chromosomes are pulled apart. The homologous chromosomes are pulled apart. Yes? So what does that look like? Metaphase? Anaphase. Spindles are pulling them in opposite directions. Yes? Understood? Does anyone have questions? Did everyone understand crossing over? Please tell me if you didn't. I'm begging you.
Can you go over it one more time?
Okay. After this, we will. Once these split, you will get two cells, one of which looks like this, and one of which looks like this. Does that look familiar, guys? That is the beginning of, basically, mitosis. But instead of having a pair of every single one, you just have one. So this is a diploid cell with 46 double-stranded chromosomes. This is a haploid cell with 23 double-stranded chromosomes. But the chromatid number here is 92, and here is still 46. Do you guys see that? Do you see the distinction I made earlier? Did it click with some of you? Yes? Just nod your heads. It's fine if you didn't. Okay, good. You saw 46 chromatids there. Why am I making that distinction? Because I'm telling you that even though you have 23, you're capable of making two cells with the entire genome from each of them. That's what I'm saying. That from here, 92, if I tell you that the whole genome is 23, you get it. 92, I can make 4 of the whole genome. 23, 46, I can make 2 of the whole genome. Because all you need for the genome is a single strand. And that's exactly what happens in meiosis 2. And the reason I never explain meiosis 2 is because it's just what? It's just mitosis. It's mitosis with one single chromosome. Take mitosis, copy it over, replicate for one chromosome. That's it. Do you guys get that?
What's the 92 part again?
Huh?
The 92?
Meaning that specific chromatids, I have 46 double-stranded chromosomes. That means I have 92 chromatids. And since in meiosis, every single cell ends up with a single chromatid of each chromosome, I'm showing you that specific thing. You look like you were crying. I'm so scared. She looked up and I just saw tears in her eyes. I was like, oh my God. This lecture really sucks. Guys, I'm going to re-explain crossing over real quick. I'm going to erase this. We're going to re-explain crossing over in a more detailed fashion. Now would be a good time to wash up for prayer if you understood that whole discussion. Okay. Everyone's good to erase this? Yeah? Sorry, I didn't ask before. Oh no, I erased the title. That's okay. I didn't want to do that. That was unintentional. That was not for the drama. When I was born, it took an intense level of precision between two people. Let's say that humans, in general, have a single chromosome. 1. Take the 23, put them into 1. Inside my mother, there are 2 pairs. There's 1 pair of that single chromosome. This chromosome she got from her mother. This chromosome she got from her father. The chromosome she got from her mother is completely black, and the chromosome she got from her father is completely white. Correct? During prophase 1 of meiosis, there is a crossing over event that leaves the chromosomes looking like this. Yes? That is inside my mother. And when these split, the homologous chromosomes will split, leaving this. Yes? And those sister chromatids will split, leaving the egg cell that created me. Let's say that this is the one with my name on it. This egg cell now contains a single version of that chromosome that is a mix between my mother's mother and my mother's father. The same thing happens inside my dad. In my dad, he has cells that are a mix of his mother and his father. Same thing happens. Crossing over in meiosis. Before anything. This makes what? And just to show you that different parts of it can cross over. Oops, sorry. Still stuck in my old ways. I'm just going to follow this one. You understand the same thing happened over there. This splits. Mix between his mom and his dad. This also has my name on it. So when these two combine, you now get a cell that formed me. And inside of my pair of chromosomes, I have a little bit from my paternal grandfather, my maternal grandmother, my maternal grandfather, and both my dad's parents from him. Yes? But now, when I have children, the same thing's going to happen with these. So now look. It's no longer as simple as one style on one chromosome. So now when you do the crossing over between 2 pairs of those, right? You'll get something that looks a little bit like this. And 4 people are packed into a single chromosome that goes on to my child, and whoever I have a child with, the same thing happens with 4 completely different people. But from those 4 people came the 4 people before them, and the 4 people before them, and the 4 people before them, and before them, and before them. And this is why every single individual in any room ever is a genetic anomaly of billions of people. Does that make sense? Does that clarify any confusion about crossing over. Do you guys see how it works? Very good. We will take a break, and we will cover the reproductive system and the menstrual cycle. Thank you guys so much for watching. Please support the channel, link in the description below. 50% of all money made off this channel actually goes towards Doctors Without Borders, right? And Zaid, can you please turn off the video for me?